Genetic variation is a fact that a biological system individual and population is different over space. It is the base of the genetic variability of different biological systems in space. Genetic variation is based on the variation in alleles of genes in a gene pool. It occurs both within and among populations, supported by individual carriers of the variant genes. Genetic variation is brought about by random mutation, which is a permanent change in the chemical structure of a gene. Among individuals within a population, genetic variation among individuals within a population can be identified at a variety of levels. It is possible to identify genetic variation from observations of phenotypic variation in either quantitative traits or discrete traits. Genetic variation can also be identified by examining variation at the level of enzymes using the process of protein electrophoresis. Polymorphic genes have more than one allele at each locus. Half of the genes that code for enzymes in insects and plants may be polymorphic, whereas polymorphisms are less common in vertebrates. Ultimately, genetic variation is caused by variation in the order of bases in the nucleotides in genes. New technology now allows scientists to directly sequence DNA, which has identified even more genetic variation than was previously detected by protein electrophoresis. Examination of DNA has shown genetic variation in both coding regions and in the non-coding intron region of genes. Genetic variation will result in phenotypic variation if variation in the order of nucleotides in the DNA sequence results in a difference in the order of amino acids in proteins coded by that DNA sequence, and if the resultant differences in amino acid sequence influence the shape, and thus the function of the enzyme. Between populations, geographic variation in genes often occurs among populations living in different locations. Geographic variation may be due to differences in selective pressures or to genetic drift. Measurement: genetic variation within a population is commonly measured as the percentage of gene loci that are polymorphic or the percentage of gene loci in individuals that are heterozygous. Sources: random mutations are the ultimate source of genetic variation. Mutations are likely to be rare and most mutations are neutral or deleterious, but in some instances the new alleles can be favored by natural selection. Polyploidy is an example of chromosomal mutation. Polyploidy is a condition wherein organisms have 3 or more sets of genetic variation. The mutation is started off by a parent. As the parent mates, the offspring now has a chance to receive that mutation trait also. Now when the mutated offspring is ready to mate, they now have the chance of passing on that trait to their offspring. This process begins the first generation of mutated offspring. Crossing over and random segregation during meiosis can result in the production of new alleles or new combinations of alleles. Furthermore, random fertilization also contributes to variation. Variation and recombination can be facilitated by transposable and transposed genetic elements, commonly known as endogenous retroviruses, lines, signs, etc. For a given genome of a multicellular organism, genetic variation may be acquired in somatic cells or inherited through the germline. Forms: genetic variation can be divided into different forms according to the size and type of genomic variation underpinning genetic change. These include small-scale sequence variation, copy number variation, copy number loss, copy number gain. Rearrangement, translocation, inversion, segmental acquired uniparental disomy. Numerical variation, polyploidy, aneuploidy. Maintenance in populations: a variety of factors maintain genetic variation in populations. Potentially harmful recessive alleles can be hidden from selection in the heterozygous individuals in populations of diploid organisms. Natural selection can also maintain genetic variation in balanced polymorphisms. Balanced polymorphisms may occur when heterozygotes are favored or when selection is frequency-dependent. See also, genetic diversity, human genetic variation, meiosis. References. Further reading, http://www.eoarth.org. Genetic Variation. McGinley, Mark. J. Emmett Duffy. 2008. Genetic Variation. In Encyclopedia of Earth. Eds. Cutler J. Cleveland. Genetic Variation in Griffiths, AJF Modern Genetic Analysis, Vol. 2, pages 7, How is genetic variation maintained in populations in Sadava, DAL. Life, The Science of Biology, pages 456. External links, Genetic Variation,